Good afternoon, everyone. My name is Guy Sherwood, and I am a 20-year survivor of Waldenstrom macroglobulinemia. Today, it is my distinct pleasure to introduce to you Dr. Stephen Treon of the Bing Center for Waldenstrom macroglobulinemia at the Dana-Farber Cancer Institute in Boston. Today, Dr. Treon will share a lot of history about the progress in research in our rare disease with a special presentation on improving outcomes through discovery, Waldenstrom's macroglobulinemia. At the conclusion of this lecture, Dr. Treon will answer as many questions from the audience that we have time for. So thank you, enjoy the lecture, and over to you, Dr. Treon.
Greetings, my name is Stephen Treon. I'm a professor of medicine at Harvard Medical School and the director of the Bing Center for Waldenstrom's macroglobulinemia at the Dana-Farber Cancer Institute in Boston. I was tasked by both Carl Harrington as well as Tom Hoffman to talk today about improving patient outcomes through discovery for Waldenstrom's macroglobulinemia. In this next slide are my disclosures. And I always like starting off my presentations by talking about Jan Waldenstrom. Jan Waldenstrom was first to describe this condition that we know today as Waldenstrom's macroglobulinemia in his publication that appeared in 1944 in Acta Medica Scandinavica. But it's important, I think, for all of us to recognize that a large gap of time actually went by before people began to recognize that Waldenstrom's macroglobulinemia was, in fact, its own pathological entity. In fact, many people at that time thought that this was a disease that was a disease much like that of multiple myeloma, except that it had, its own intricacies. And so this is back now in 1943, 1944 that he describes Waldenstrom's macroglobulinemia. And it actually wasn't until 1950 that Tischendorf made the insight that mast cells were being found in excess numbers in the bone marrows of patients with Waldenstrom's macroglobulinemia. That actually was very pivotal because it was the first recognition that there was this pathological entity associated with what Jan Waldenstrom had described. In 1962, the Massari brothers then described a familial form of Waldenstrom's where multiple siblings actually had macroglobulinemia. And as you can see, as the disease became recognized, then a number of therapeutic modalities were introduced, new drugs, including chlorambucil being first introduced in 1961. In 1978 came along the understanding that IgM-MGUS was actually a predisposition for Waldenstrom's macroglobulinemia, and my wonderful mentor and colleague Bob Kyle made this observation. And then we began to see the nucleoside analogs, predominantly being developed at the MD Anderson, initially by Hago Kantarjian, but then my other friend and colleague, Thanos Demopoulos, who then also helped develop 2-CDA, which is another nucleoside analog. And of course, my friend and colleague, Veronique Leblond, expanded this understanding of the role of fludarabine through studies done in France as part of the French co-op studies in the late 1990s. And it was about this time that we had made the important observation that rituximab, the monoclonal antibody, was active in Waldenstrom's macroglobulinemia. And this, of course, heralded a number of other studies that we'll talk a little bit about in just a moment, combining with rituximab with a number of many of the agents that are used today. But I think it is important to also recognize that despite the fact that we're moving along in the development of new drugs for this disease, we still lacked no published diagnostic criteria for what was Waldenstrom's macroglobulinemia. In addition, we had no criteria to treat. So there were many times patients who didn't require treatment, who were asymptomatic, that were being treated. In addition, there were no treatment guidelines. So there was no guidance that existed out there on how to go about treating a patient with Waldenstrom's macroglobulinemia. And even perhaps, more to the reality, we didn't even have response criteria. We didn't really know how to categorize how a patient did after receiving drug therapy. And this is why what Judith May and Bruce Chesson did in 2000 was very, very important. And that was bringing together experts for the first international workshop on Waldenstrom's macroglobulinemia that occurred in Washington, D.C. And this was the first time that experts actually got together to talk about this disease. Now, what I will tell you ended up happening at the end of that meeting was the fact that we all agreed that these were the things that we were lacking: diagnostic criteria, criteria to treat, treatment guidelines, response criteria. And I think we also agreed that we needed to work together to be able to act on these very important omissions that existed up to that point in time. Now, I think many of you are going to recognize Arnie Smokler in this picture, the founder of the IWMF. And Arnie was a wonderful friend who I would spend countless hours on the phone talking to him about macroglobulinemia. He was very supportive of the work that I had embarked on at that time, which was to develop and expand antibody-based therapies for treating Waldenstrom's patients. And I was the fortunate recipient in January of 2000 to receive a grant from the IWMF, which you see here, which was to look at treatments of Waldenstrom's by antibody-treated mediated immunotherapy and induction of tumor-selective antigens. And this actually was a very important grant because it gave me the opportunity to be able to study the role of Rituxan as well as other monoclonal antibodies for treating this disease. Now, as I alluded to, a lot happened soon after we began to recognize the activity of rituximab in Waldenstrom's macroglobulinemia. We began to conduct a number of other studies, which included studies looking at the role of CHOP plus Rituxan, the role of extended Rituxan. We also looked at thalidomide and lenalidomide combinations with Rituxan. We also combined fludarabine and Rituxan in a large international study. We were the first to report on the role of Velcade along with Velcade, dexamethasone, and rituximab. And soon thereafter, we also published our studies on alemtuzumab, which is also known as Campath. We were the first to actually publish on the role of bendamustine and rituximab in combination. We also published the first study on maintenance rituximab. And then came the study of pomalidomide, dexamethasone, and Rituxan. And throughout these years, going from 1999 all the way to 2012, we also made some very interesting observations, including that Rituxan could cause the IgM flare, which we published back in 2004. In 2005, we also found that patients actually had a genetic background that determined how well they responded to rituximab. These were the Fc gamma R3A polymorphisms. And then, of course, in 2011, we also published on the importance of achieving at least a very good partial response in order to predict long-term response to rituximab-based therapy. Throughout this entire period of time, as we developed these new therapeutic modalities, the IWMF was a wonderful partner in being able to fund and support these very important and critical clinical studies and clinical observations. And I just wanted to share with you some photos because this will actually show the wonderful relationship that the Bing Center had with the IWMF. Here we're, at the IWMF meeting in 2002. We see here my wonderful friend, Ben Rood, who was the president at the time. But what we had done at that meeting was we actually organized a clinic where we were actually drawing blood from the patients and we were looking at their medical history and their outcome with certain therapeutics. And this was very important because it was really those blood draws and our ability to do the genomics that made us understand how important the polymorphisms in the Fc gamma R3A receptor were to actually forecasting how a patient would do with rituximab-based therapy. Now, given the fact that we still had a lot of unanswered questions and there was a necessity for us to continue the workshop process, we organized in 2002 the second international workshop on Waldenstrom's macroglobulinemia. The IWMF was very, very pivotal in the organization of this workshop in Athens, Greece. And here I'm actually showing this very nice picture of Ben Rood. And these are pictures from the closing ceremony, which was took place at the Acropolis in Athens. You can see, in fact, that we had a wonderful turnout for this meeting. 56 investigators who represented multiple countries came together. And I had the privilege of organizing this meeting along with Bob Kyle and Thanos Demopoulos. And you can see here in the screen the small room where we all sat for several days as we went about developing the important criteria that really even today are pivot points in the treatment and management of patients with Waldenstrom's macroglobulinemia. And these were published in seminars in oncology and included the diagnostic criteria for Waldenstrom, the criteria to initiate treatment, treatment guidelines, as well as response criteria for Waldenstrom's macroglobulinemia. Now, of course, the Waldenstrom's workshops have continued since that time. What is wonderful to just share with you is how these workshops have continued to grow. This is the third international workshop, which took place in Paris in 2004. And you can see that the family of investigators grew during this period of time. And this was a meeting that I helped organize along with my colleagues Jean-Paul Fremond, Véronique Leblond, as well as Pierre Morel. In this next slide, we see that the family continues to grow. This is the fifth international workshop on Waldenstrom's macroglobulinemia that took place actually in Stockholm, Sweden. And this was our closing ceremonies in the Nobel Hall, where we had the opportunity to honor the wonderful work of Jan Waldenstrom, and we had many members of the Nobel Committee that were actually present during this time. Now, in addition, I think, to the development of new therapeutics for Waldenstrom's, there's also been this very important legacy of recognizing that many of the drugs that we had been using to treat Waldenstrom's actually could cause other problems, including other cancers. And this was first recognized, in a paper that we published in the Journal of Clinical Oncology in 2008. My colleague Xavier Lalou was the first author, along with Jake Sumarai. And here we followed 463 patients with Waldenstrom, and we looked to see if there were differences in patients who were receiving fludarabine or cladribine compared to patients being treated with other modalities for Waldenstrom's disease. And what we came to learn in this study was that the risk of transforming to an aggressive lymphoma went up by 7-fold, and the risk of having either myelodysplasia or acute leukemia, myelodysplasia being a potential precursor to acute leukemia, went up by 3-fold in patients treated with a nucleoside analog. Important to note in this study that those patients that did transform to an aggressive lymphoma were often salvaged with the use of therapy such as CHOP Rituxan, which we typically use to treat patients who have these aggressive lymphomas. But this was a very important study because it began to make us think about the safety, long-term safety of the therapeutics that we use. This was also a very important study that we published in 2009, where we looked at CHOP Rituxan, which really was one of the mainstays of therapy in its time for patients with various lymphomas. Today, it continues to be a mainstay of therapy for aggressive lymphomas. But what we were interested in knowing is, do we really need an aggressive regimen like CHOP Rituxan when in fact, other regimens that don't have adriamycin, which is a very important component of this therapy, which can cause a lot of toxicity, as well as vincristine, which is the V that you see there, which causes neuropathy. And so we wanted to see how patients would do with just CPR, which was cyclophosphamide, prednisone, and Rituxan, and not really, needing either the V and vincristine or the O, which is the drug Oncovin or adriamycin. And what you see here is, in fact, that even simplifying therapy, the response rates were the same, including major responses. And so I think this was a very important study because it showed us that we could get away with less and have, less toxicity, less neutropenia, hospitalizations, as well as neuropathy due to the vincristine that we used in both CHOP as well as CVPR. Now, at that time, we also recognized that we were just borrowing therapies from other disease states, whether it was our myeloma colleagues or CLL colleagues or lymphoma colleagues. We were waiting for them to develop therapies, and then basically these were handed down to us. So most of what we were using were hand-me-down therapies. So in Los Angeles in 2003, we brought together this very important brain trust of basic scientists and translational scientists. This is the meeting that we had as part of the patient summit at UCLA. And what we all felt was critical to our ability to be able to innovate and move the field forward for Waldenstrom's was that we needed a center of excellence to study this disease, to be able to do the basic science, to understand what the genomics were that were driving this disease. And so this was really a very pivotal moment. And of course, in 2005, we were able to have the center that we dreamed about, and that was the Bing Center for Waldenstrom's. This was made possible by the generous funding by Dr. Peter Bing. And this here, what you're seeing here is the dedication ceremonies where Harriet Fulbright, who was then the president of the Fulbright Foundation, joined our president, Edward Benz, for the dedication of this center. And here you can see the ribbon cutting with Dr. Peter Bing, who himself is a physician, but also was a trustee and chair of Stanford University at the time, cutting the ribbon that dedicated this Center of Excellence for Waldenstrom's macroglobulinemia at the Dana-Farber Cancer Institute. And with this center, we were able to recruit a wonderful brain trust. And this included both doctors as well as scientists, people from the United States as well as abroad. And we had the ability to work together to be able to understand what the disease was at the basic science level, but also to understand how many of the drugs that we were using were impacting patients and what we needed to do to be able to innovate and move the field forward. Now that was very nicely accomplished in 2011 when we reported at the American Society of Hematology meeting the discovery of the NYD88 mutation, which was made possible through whole genome sequencing. And this was an effort that was funded by both Dr. Peter Bing, but also by the IWMF. Both entities provided the critical funding that paid for whole genome sequencing, which at that time, it's very important to recognize, was still a very new art. Very few people had used it, and it was very expensive. So being able to have the IWMF and Dr. Bing pay for this and provide this critical funding, where 30 patients underwent whole genome sequencing was very critical. And what we discovered as part of this effort was information that we're still using today. But critical among this was the fact that 93 to 97% of patients with Waldenstrom's macroglobulinemia have actually a mutation in the NYD88 gene. And here shown is the whole genome sequencing results that actually show the changes that we detected in the DNA of patients who had Waldenstrom's macroglobulinemia that were consistent with this recurring mutation in NYD88. Now, why this was a pivotal discovery was that, for the first thing is it gave us a test that we could use to actually help us distinguish Waldenstrom from other things that secrete IgM and look like Waldenstroms, including marginal zone lymphomas, multiple myeloma, certain CLLs. We did find MYD88 being expressed in patients that had IgM MGUS, the precursor condition. And the stronger the signal was, the more that it determined or predicted that a patient would go on and develop Waldenstrom's macroglobulinemia. And here what you're seeing is PCR analysis, where the stronger the signal is, the earlier you're detected. And so even at lower levels of PCR cycling, you're able to pick this up. And that's why the Waldenstrom's patients are all showing up, here at the bottom. And with this PCR technique that we developed, 93% of patients with Waldenstrom were positive. Important to note, the PCR technology that we developed, thanks to our colleague Leanne Zhu, is still used in most medical centers throughout the United States as well as abroad, because she had developed this very, very intricate test. Now came along Dr. Guang Yang in our group, who has been instrumental in being able to tell us what exactly NYD88 does. What he found was that this was a protein that when it became mutated, it actually turned on BTK, Bruton's tyrosine kinase, which is the target of Ibrutinib, as well as other BTK inhibitors like Zanubrutinib, Acalabrutinib, as well as Tirabrutinib. He also made the very interesting discovery that some of these BTK inhibitors, like Ibrutinib, and now we know also Zanubrutinib, also silenced HCK, and HCK is one of those molecules that's actually turned on by mutated MYD88. And this is actually the linchpin that turns on BTK, as well as other survival pathways like ERK and AKT that actually turn on the cell's ability to grow and survive. And more recently, with my other colleague, Dr. Maneet Munshi in our group, he also discovered that this other molecule, SYK, that you see here, is also turned on and can also turn on other pro-survival pathways. And this is relevant because there are drugs that can actually silence out SYK itself. And so having this very important diagram to understand how MYD88 functions has been critical to the development of many new therapeutics for Waldenstrom's macroglobulinemia. Now, in this next slide, I show you also the important discovery of yet a second mutation. This was also made possible by the whole genome sequencing project, but involves CXCR4. This is a mutation which I'll tell you about in just a second, but it was made possible because of the wonderful work by Dr. Zachary Hunter in our group, who went back and analyzed the whole genome data, but he created his own algorithms that allowed him to understand that there were these mutations in CXCR4 affecting about 30 to 40% of all Waldenstrom patients. Now, CXCR4 is actually a receptor. I'll show you in just a moment what exactly it does. But almost all the mutations that we found in patients with Waldenstroms all clustered in that little pigtail that you see at the bottom, including the S338X mutation, which is found in about half of all patients who have CXCR4 mutations. And that's critical because what that mutation does, it actually causes a stop to be introduced, and that whole tail that you see after S338X actually just gets cut off, it gets lobbed off. And that's really important because this part of the tail of this gene is very critical for actually regulating how this gene performs. I'm going to show you in just a second what exactly I mean by that. I also want to tell you that in addition to the S338X mutation that we found, there have been a number of other mutations. In fact, now we have over 40 types of CXCR4 mutations that we have cataloged. And this includes the type of mutations that I mentioned with the X that actually cut off part of the protein. But also there are mutations known as frameshift mutations where DNA is either inserted or omitted as a result of the mutation. And this scrambles the code and it makes actually the whole protein very, very different. Now, why this is important is because when you look at how a patient presents with Waldenstrom's macroglobulinemia, the type of mutation they have can be very, very instructive. if you have the L265P MYD88 mutation and you have the S338X nonsense mutation, you're going to be the type of patient that's going to come in with a very high IgM and maybe even hyperviscosity crisis. If, on the other hand, you have no MYD88 mutation, it may be that you're one of those patients that's going to come in with very low disease burden. So this was a very important study because it showed us how we could use this mutation information to actually see how a patient would present. Now, I mentioned to you CXCR4 is important. And why is it important? Because it's a receptor for CXCL12. This is a protein released by the bone marrow stroma, and it actually causes the Waldenstrom cells to want to go to the bone marrow. This is the reason why we believe that we see this trafficking of Waldenstrom cells to the bone marrow. And when you have a CXCR4 mutation, the antenna that actually signals for CXCL12 remains in the on position and continues to signal. And as a result of that, it actually upregulates a number of proteins and activates them, I should say, that actually produces drug resistance against a number of drugs, including the BTK inhibitors. And that's why the kind of modeling that we and others have done around CXCR4 has been very, very instructive. Now, with that information in mind, we were very eager to test BTK inhibitors in Waldenstrom's. We conducted this multi-center study of Ibrutinib in relapsed and refractory Waldenstrom patients. Refractory means that the patients didn't respond to their prior therapy. Relapse meant that the patient had progressed after being treated and needed therapy. And patients had to have at least one prior therapy. And so with Ranjan Advani at Stanford and Leah Palumba at Memorial Sloan Kettering, we launched this study treating patients with Ibrutinib at the dose of 420 milligrams a day. And we would continue treatment as long as the patients didn't progress or had unacceptable toxicity. And as part of the study, we actually looked at their MYD88 and CXCR4 mutation status. Now, this was a very important study for two reasons. One, it received the first ever breakthrough designation in any oncology indication. And I think we ought to be proud of Waldenstrom's to have been able to achieve that. This was achieved in November of 2012 when we presented both our scientific data as well as the early clinical data that we had on Ibrutinib to the FDA. But then, of course, came the very important discovery in 2014 of the activity of Ibrutinib in Waldenstrom's disease. And this led to the first ever approval of a drug for the treatment of Waldenstrom's macroglobulinemia. This is the FDA meeting we had in June of 2014. And of course, as many of you know, in January of 2015, the approval by the FDA came along. And then later in 2015, by the EMA. Now, I just want to update you, since we have the opportunity today to talk about the BTK inhibitors, on the updated activity of Ibrutinib in previously treated patients. This is an update of that pivotal trial, now with a median follow-up of 59 months. And I think what the takeaways here are that 90% of those patients responded, even though the patients had a median of 2 prior therapies. About 80% had a major response, and 30% had a very good partial response, which means at least a 90% decrease in a disease burden. And important here also was the fact that depending on what the patient's genotype was, this impacted how they did. If they had no MYD88 mutation, we didn't see really any major responses or VGPRs. If they had the MYD88 mutation and also had on top of that a CXCR4 mutation, you see that the response rates were actually, major response rates are good, but not as good as those patients that have only MYD88 mutation. And this was also true of the VGPRs, the very good partial responses, where you can see there the big difference. 47% got a VGPR who only had a MYD88 mutation versus 9% who had both the MYD88 and CXCR4 mutation. And also important, it took longer to get to a major response, almost by 3 months with this update. So you have both MYD88 and CXCR4, it just takes longer to get to that response. That may be critical in some situations, and we'll talk about that at the end of my talk. Also, some good news to share is progression-free survival, how long the patients actually had their disease controlled by Ibrutinib. At 5 years, 54% still remain without progression of their disease, and 87% of the patients were alive. But you can see there are differences in how the patients progressed depending on their mutation status, with NYD88 mutated only patients showing the best outcomes. 75% were free of progression at 5 years. Those patients that had both mutations, the progression-free survival was about 4 years. So that's actually very good when you consider all the other options. And the patients that had no NYD88 mutation, showed, the earliest progression. At the update of this study, we also looked at long-term toxicity. There were more atrial fibrillation events, now 12.7% with the longer follow-up, since we first reported this study in the New England Journal of Medicine in 2015. But I think what's also important to say is that 7 out of 8 of these patients continued on the Ibrutinib with medical management. And so developing the atrial fibrillation didn't alter treatment for the vast majority of these patients. Now, since that pivotal trial, we also have learned about the Innovate study. This is a very interesting study where patients got Ibrutinib and Rituxan or a placebo plus Rituxan. And I want to just emphasize that even in this very important international study, randomized study, which showed that Ibrutinib and Rituxan did better than placebo and Rituxan, that even among those patients who had the NYDA mutation plus CXCR4, there were fewer major responses, fewer VGPR responses, but the time to get into that response was about the same as those patients that had only the NYDA mutation. And so this is an important, lesson to come by. Now, we also learned in this study, which was updated by Christian Buskey at the ASH 2018 meeting, that the patients that had also the CXCR4 mutation progressed earlier than those that didn't. And so there was yet another study that was showing us that the patients who have CXCR4 mutations tend to progress earlier on Ibrutinib-based therapy. Now, I think one of the proud studies that we published, and this has been expanded on by Jorge Castillo in our group, was the role of Ibrutinib in patients having Neal syndrome. This is, of course, when the CNS or the brain and the spinal cord become involved with Waldenstrom's disease. And this used to be so difficult to treat. And so with my colleague Steve Allen, who's down in New York, we published this study on a mutual patient that we treated with Ibrutinib at the dose of 560 milligrams per day. And this patient actually had a very bad case of being Neal syndrome, as you can see here, both by MRI, but also by PET scan, where you see the yellow area really representing very metabolically active disease. But you can see just a few months later how much has changed with this patient being on Ibrutinib. And we were able to also show that we had meaningful drug levels getting into the brain, into the CSF of the brain. This was a major game changer for being Neal syndrome patients, because prior to that, we had to admit the patients and treat them with high-dose chemotherapy, and often we would end up with a lot of toxicity. Now, the ability to actually innovate around the development of BTK inhibitors has been very important. We now have some new drugs. I know Jorge Castillo has already talked to you about Acalabrutinib and Zanubrutinib being new introductions to the treatment of Waldenstrom's. I think it's important to keep in mind that there are differences in these drugs because of other targets that they can affect. And this, of course, can affect, the outcome in terms of side effect profiling. Here in the Acalabrutinib study that was published by Roger Owen this past year, they found an atrial fibrillation rate of 5%, although their follow-up is much shorter than the one that I reported. And close to that time, our atrial fibrillation rate was also about 5%. But keep in mind, no atrial fibrillation event led to Acalabrutinib being withheld or discontinued here. Headaches were very common in the beginning of treating patients with Acalabrutinib, but these very quickly resolved within a few months' time. Now, the big trial that I know Jorge has already talked to you about is the Aspen trial. This has been this year's big news. It's a trial where zanubrutinib was compared to ibrutinib head-on. The follow-up time here is relatively short, about 19 months, but there were some very important lessons that came out of this study, including the big difference in side effect profile with considerably less atrial fibrillation being seen in patients treated with zanubrutinib, less diarrhea, less hemorrhage, less hypertension, but conversely, more neutropenia where the white blood count, goes down. However, there were no opportunistic infections which were seen with this, neutropenia among zanubrutinib patients. And so I think at the end of the day, we're going to learn about how we're going to use each and every one of these BTK inhibitors to treat Waldenstrom patients based on their efficacy, which seems to be about a similar level at this point in time, but also looking at the side effect profile. And there may be unique areas, where one drug may be preferred over another. For instance, we have a lot of experience now with Bing-Neal syndrome with ibrutinib. This may be, a patient population where we still want to prioritize the treatment of Bing-Neal syndrome with ibrutinib. Conversely, we saw that, as Jorge has also mentioned to you, more VGPRs and deeper IgM reductions with zanubrutinib. And that may be important in situations where a patient has disease that is being impacted by the IgM, like the neuropathies, for instance. And so this is why the ability to have multiple BTK inhibitors is very critical to our field. And I think BTK inhibitors are here to stay, and that's why strategies to enhance BTK inhibitors become all the more important. As I already mentioned to you, CXCR4 remains a very important problem. Those patients that have the CXCR4 mutation tend to have less deep responses. We know this also to be true with the zanubrutinib study. And it takes sometimes longer to get into those deeper responses, as we learned also with the ibrutinib study. And so why not go after CXCR4? And here the Leukemia and Lymphoma Society has been very important to us because they helped us fund this study with ulocuplimab, which is an antibody that blocks CXCR4 signaling. And the results from the study are actually very positive. We're going to hopefully report these very shortly at the American Society of Hematology meeting. But we'll have maybe some chance during our discussion to maybe talk a little bit more about this study. Right now, we are treating patients with another CXCR4 inhibitor, and that is the drug Mulvarexophore. This is an inhibitor which actually you take by mouth, whereas ulocuplimab is an antibody that you have to get by intravenous injection. And so here we have the opportunity of perhaps providing all oral therapy. We get, a drug like ibrutinib, and you also get Mulvarexophore as an oral inhibitor. And this trial just has started. It's a very important trial. This is the phase 1/2 part of the trial, but this will be followed by a randomized trial where patients will get ibrutinib or ibrutinib plus Mulvarexophore who have the CXCR4 mutation and require therapy. Now, I also am very proud about this work that we published back in 2015 involving the drug Venetoclax. At that time, it was known as ABT-199. We were the first to actually show that BCL2 was critical to how ibrutinib functioned in cells that had the NYDA88 mutation. And these are Waldenstrom cells that you see here. And what you see where the arrows are shown, the first set of arrows where it says ABT and IB, is that in cells that were either had no CXCR4 mutation, those wild type cells, or those that had CXCR4 mutation, the S338X that we engineered, that if you added both drugs together, you actually caused all these apoptosis markers of cell death, to go up dramatically over each drug, individually. And this was very important because we know that BCL2, which is what Venetoclax targets, is highly expressed in Waldenstrom cells. And this is an anti-apoptosis protein. It actually defends the cells from dying against drug insult. And so if you can knock down, a protein that's actually defending those cells and add another drug that's actually promoting death, then as we see here, we have the ability to kill those cells off. And that's why the study that Jorge Castillo is now doing in our group, combining Ibrutinib and Venetoclax, is very, very critical. We hope that this will lead to deeper responses in patients with Waldenstrom, but also this treatment program is for 2 years, and then drugs all stop. Instead of being able to give drug, for long periods of time, until progression, we have perhaps the opportunity to only treat patients for 2 years' time and then stop. And that's why I think this is a very critical study, and we're really looking forward to the outcome. Now, another very important study that the IWMF has funded is to look at resistance mechanisms to Ibrutinib. And these are 2 critical papers I want to show you where the IWMF support is strongly acknowledged. And let me tell you at least some of the lessons that we learned from this study are now being purposed in other diseases. So it's wonderful when you got an orphan disease like Waldenstrom being able to teach people in other disease groups valuable lessons. And so let's talk a little bit about resistance. First of all, there is this mutation in BTK that was discovered by Lian Zhu in Waldenstrom patients, which is in the amino acid cysteine 481. This is important to remember because that's where Ibrutinib binds to BTK. And in many patients, we found actually mutations in cysteine 481 that converted that cysteine to some other amino acid, and Ibrutinib no longer was able to bind. And we saw, in fact, in some Waldenstrom's patients, they not only had one type of cysteine mutation, they had multiple different types. So you had all these different resistant clones emerging in these Ibrutinib-treated cells. Now comes the very exciting science, and that is that when we saw in the Waldenstrom patients that had the BTK cis mutations, it usually was always a small minority of the cells that had the actual mutation, and yet the patients were progressing on Ibrutinib. And my colleagues who work in CLL saw the same exact thing, and so there was always this mystery as to what was going on. And so Jiaji Chen in our group, along with his mentor Guang Yang, did these very elaborate experiments. They use what we call co-culture dishes, where you can actually put cells on both sides of a membrane and see what happens. And when they put cells that had no BTK mutation, ones that are shown in green, along with the yellow cells that also had no BTK mutation, and they added Ibrutinib, everything died. But if they engineered the cells to express that BTK mutation, the CYS481 mutation, and they put it on one side, the red ones here, and then they added Ibrutinib, they all lived, including the cells that didn't have it. And so there was something going through the porous membrane that was making the cells on the other side of the membrane resistant to Ibrutinib. And what we discovered was that interleukin 6 and interleukin 10 was actually being shed by those cells that actually created resistance in the cells that didn't actually have the mutation. And that was a very important discovery because we were able to work out the pathway, the ERK pathway was identified, and we were able to show that if you did in fact use a blocking drug to ERK, you could actually reverse this resistance mechanism. These days, we've embarked on also looking at non-covalent BTK inhibitors. These are drugs that actually bind to BTK, but at sites other than the BTK CYS481 site. And at our institution, we're working on LOXO305 that actually targets BTK at G473 and K483. So these are very removed sites from the cysteine 481 that is responsible for resistance to Ibrutinib. That same site, by the way, is the same site that Zanubrutinib, Acalabrutinib, and Tirabrutinib bind. So that mutation would also affect those drugs. So having these non-covalent inhibitors being developed is very important and gives us a chance to actually continue to treat patients who have a BTK CYS481 mutation with BTK inhibition. Now, the IWMF has also funded a very important study, which is looking at the development of inhibitors that target HCK itself. As you remember, I said earlier that HCK is actually turned on, it's upregulated by NYD88, and in turn, HCK is what actually turns on BTK itself. So if you can actually target HCK, you might be able to shut down BTK, but also shut down the other pro-survival pathways, ERK as well as AKT. ERK actually is the one that releases IL-6 and other inflammatory cytokines. And so if you can actually shut down HCK, you might get BTK, but also all these other very important survival and inflammatory pathways. And so with the IWMF's support, we did in fact develop with our Harvard medicinal chemistry colleagues a lead molecule known as KIN8194. We just presented this data at the American Society of Hematology meeting in 2019. And what I'll tell you what was very exciting is we actually put NYD88 mutated tumor cells into mice. We either gave them a placebo control, we gave them Ibrutinib, or we gave them KIN8194. And what you'll see there is that the animals that got the vehicle control died, very, very quickly within a few weeks of time, and their tumors grew. The animals that were on Ibrutinib actually did have extended survival, but these tumors also began to grow, and the animals ultimately started dying off a few weeks after that. And then what we show here is that KIN8194 animals actually had suppression of their tumor and showed the best survival. And in fact, in about half of the mice that were treated in this study, these mice, even after the drug was stopped, went on for another 12 weeks without developing any tumor. And so operationally, they were actually cured. Now, there's also this model that I'm very excited to share with you, where we actually put in the BTK cis mutation, the one that creates the resistance to Ibrutinib. And what we saw here was that the animals that got treated with Ibrutinib or the vehicle control, all their tumors grew very, very fast. But when you treated the animals with either the KIN8194, either at 50 milligrams or 75 milligrams, you were able to suppress the tumor growth, particularly at the higher dose. And this resulted in extended survival for these mice by many, many weeks. And so we were able to show that we could overcome the resistance associated with the BTK CYS481 mutation, which is responsible for Ibrutinib resistance. In the last couple of slides, I just want to talk about, Waldenstrom's, where we have not been able to demonstrate a NYD88 mutation. Thanks to IWMF funding, we embarked on doing whole exome sequencing that actually identified a number of very interesting new mutations in these cells, including TL1, XR1, as well as other mutations that activate the NF-kappa B pathway. But surprisingly, when you look at how these mutations actually transactivate the cell, this is what we call principal component analysis, where we take the 500 highest, expressing genes. And what we saw, in fact, was that their pattern of expression very much overlapped those of patients with the NYD88 mutation. This, of course, one would expect because, after all, this is Waldenstrom's disease. But many of these other types of mutations actually have a way of what we call phenocopying, or acting similar to what we see as part of the NYD88 signaling cascade. Now, the one thing to keep in mind, and this explains why we don't see those deep responses with Ibrutinib in patients that have no NYD88 mutation, is that almost all of these mutations are below BTK. That's where you see the little lightning bolt. That's where those mutations have been discovered. So at least we have an understanding of both the mutations responsible for NYD88 wild type disease, again, no mutation, so we call it wild type. And we also begin to understand why BTK inhibitors may have a limited role in treating these patients. I do want to share with you also where the IWMF has been very supportive, along with the Waldenstrom's Macroglobulinemia Foundation of Canada, and that is in supporting the 300 project. This is a very ambitious project which we began in 2007. The aim of this project was to sequence and track 300 symptomatic, untreated Waldenstrom's patients. And in doing so, we were determined to map out their DNA, RNA, as well as what we call the epigenome, all the regulatory machinery that helps control how DNA codes for RNA. And we wanted to understand the impact on the disease presentation, the course of disease, as well as survival of these patients, and also hoping to develop new targeted therapies based on the mutation profiling from individual patients, and their genomic mutations. Lastly, I just want to leave with you the notion that these genomic endeavors are helping us not only in developing new treatment strategies, but also in being able to make specific decisions about treatment for individual patients. And so when a patient does present and has just a NYDA mutation, we'll prioritize a BTK inhibitor, because as we saw in the data from the pivotal study, these patients actually do quite well for long periods of time. If they have both mutations, we're going to want to learn if the CXCR4 mutation is causing changes such as hyperviscosity that need immediate therapy, in which case we'll prioritize bendamustine or a proteasome inhibitor. But if they're in no rush, then we'll give a BTK inhibitor plus Rituxan, because that addition of Rituxan causes earlier responses to occur. And if they have no NYDA88 mutation, then we'll prioritize either bendamustine or a proteasome inhibitor. And because we know that BTK inhibitors, at least Ibrutinib from our own studies, has a limited role. And the same is also true of patients who have relapsed or are refractory to their prior therapy. We can use a similar schema to make informed treatment decisions. So at the end of the day, the genomic discoveries in Waldenstrom's, which we are grateful to the IWMF for their support in being able to make these discoveries, to translate them, to look at them in clinical practice, has given us the opportunity to now advance not only new drugs, such as Ibrutinib and Zanubrutinib, Acalabrutinib, Ibrutinib, but also the CXCR4 inhibitors, and our ability to also leverage drugs like Venetoclax along with these BTK inhibitors. And at the end of the day, not only will we have new drugs, but we'll also have an algorithm for customizing or personalizing the individual care of patients with Waldenstrom's macroglobulinemia. And with that, I'd like to thank the IWMF for having me in this year's forum, and I look forward to speaking with you during the question and answer session. Thank you very much.
Thank you very much, Dr. Treon, for this interesting perspective on the history of this rare disease and for reviewing the progress of research in Waldenstrom's. I think that all of us affected in one form or another by WM are truly appreciative of the enormous efforts made by you and your very capable colleagues at the Bing Center for Waldenstrom's macroglobulinemia at the Dana-Farber Cancer Center. And in fact, we are all appreciative of all the talented and dedicated researchers worldwide. Now, we're going to have a few questions from the audience for Dr. Treon. And in fact, our first question, if Dr. Treon is ready, is, has there been... Oh, thank you, Steve. Nice to hear your lovely voice. Has there been any research on delaying treatment until symptoms warrant versus treatment of increasing IgM numbers?
Yeah, it's a great question. So, we have looked at therapies that were meant to keep patients from progressing. So those studies were really focused on delaying progression of disease so that patients didn't really warrant, more intense therapy. They included some drugs that, like Viagra. They included sildenafil, that's Viagra. They included simvastatin, which is a drug that affects, cholesterol. These had all been shown to have some potential activity. We have also done preclinical studies that have looked at components of green tea, EGCG specifically. So there have been actually studies looking at therapeutics that could prevent progression. None of these, I have to say at this point, were shown to be, effective, or at least not effective for long periods of time. Therefore, I really wouldn't put any value in making any decisions based on these attempts. But, to the question that's been asked, yes, research has been done. Now, the other question about increasing IgM is very important because typically we don't treat patients based on the IgM advancing in and of itself. Usually we want to couple that with symptoms or a drop in hemoglobin as opposed to looking at the IgM per se, and that's why it's not in our official consensus panel recommendations for starting treatment. However, that being said, typically when patients are at 6,000 milligrams per deciliter or higher, there is a much higher risk of hyperviscosity crisis, and therefore, based on the work that's been done predominantly at our institution, we would advocate for treatment of somebody whose IgM may be at 6,000 or higher. Now, don't take these numbers as absolute, but just know that in that range we become very, very concerned, and we just don't want somebody to present with loss of vision as their first, manifestation of high IgM level.
Okay, thank you very much. That's a great answer to a great question. The next question is there a medication or a treatment, I suppose we could say, for Waldenstrom patients with CXCR4 mutation, since Ibrutinib does not work so well in this particular case?
Yeah, this is a great question. I think we addressed it a little bit in my presentation, but we just concluded a study looking at the monoclonal antibody ulocuplimab. So it's an antibody like Rituxan. It's able to bind to CXCR4, and it prevents CXCR4 from engaging with the protein that normally turns it on. That trial actually is a very positive one. We were very excited by the readout. We showed we could get patients much quicker into a response, even deeper responses. Unfortunately, the sponsor of that trial, the pharmaceutical company, has decided to discontinue the manufacture of ulocuplimab. That's because they were really targeting it for treating patients with acute leukemia, and they didn't get a signal there. And for a rare disease like Waldenstrom's, they just didn't believe that they could make the case to continue its development. But in its place, there's a pharmaceutical company here in Boston called X4 Pharmaceuticals, which has actually developed an oral antagonist or blocker of CXCR4. It goes by the name of Mulvarexophore. It's taken by mouth once a day, and we are running this trial now. So we're very excited about this trial because, quite honestly, it's an innovation. Beyond having to have the patient come into the clinic to get an antibody, this way you send the patient home with Ibrutinib and Mulvarexophore. And so I think this is going to be a very exciting trial. And if anybody is interested in this trial, we just actually started it very recently. You have to have CXCR4 and you have to, need treatment. And the patients will get both Ibrutinib as well as